Explore the history, relationships, expertise, and data that go into ensuring Stine growers get maximum yield potential. This is the Stine Seedcast. Here's your host, David Thompson.
Hello, and welcome to the Stine Seedcast. I'm your host, David Thompson, National Marketing and Sales Director for Stine Seed Company. We've got another great episode lined up with special guests, expert insights, and discussion on everything you need to know about maximizing yield potential. Today, I'm excited to welcome Dr. Greg Tylka to the show. Dr. Tylka is the director of the Iowa Soybean Research Center and a Morrill professor in the Department of Plant Pathology and Microbiology at Iowa State University. Thanks for joining us today, Dr. Tylka.
Thank you, David, for having me.
So Dr. Tylka has spent years studying the biology of soybean cyst nematodes and their effects on soybeans. His current research includes the study of SCN-resistant soybean varieties and effects of nematode-protected seed treatments and interactions between SCN and insect herbivores. Soybean cyst nematodes are an ever-present problem for soybean growers, causing billions of dollars of yield destruction each and every year. We're going to hear from Dr. Tylka on the evolution of these pests and how growers can manage their presence now and in the future. So let's get started. So first off, a bit of a disclaimer. So, Greg, you and I have known each other for a long time.
Yep, I believe I was your first interview back in college.
Probably.
Yep.
And then my girlfriend at the time, who became my wife, took her master's under Greg at Iowa State. So like I said, I think I've known you for 30 years. Yep. But I always like to start our interviews with a bit of context. Mm-hmm. So tell us a little bit. First of all, you're not actually from Iowa originally. So tell me about your path to the Midwest.
Well, I was born and raised in southwestern Pennsylvania near Pittsburgh, about 20 miles away from Pittsburgh. I grew up along a river. I've always been in love with biology. I'm embarrassed to say I used to shoot bullfrogs with my BB gun and then cut them open to see what makes them tick inside. And then I developed this weird ambition of being a marine biologist. And I wanted to train sea animals at SeaWorld. Oh. But came from a relatively poor family, couldn't afford to go to an out-of-state school. It's hard to study marine biology in the southwest corner of Pennsylvania. So I went to a local school, state-run university called California University of Pennsylvania and got a biology degree.
Okay.
When I got finished with that, I couldn't find a decent job. And the professor who I was washing glassware for to make some money heard I had done good in my classes, and so he offered me money to work on a research project and get an assistantship. $250 a month I got paid, and plus free school. And I did a project studying fungi that rotted pine seedlings. So that was a 2-year stint, at the end of which I had a master's degree in biology. And then I still couldn't find a good job. And that professor said I'd done so well, he advised me to go on for a PhD. And that took me to the University of Georgia. And when I got to Georgia, I was able to work with any professor I wanted to. I picked a professor that worked on nematodes. And most of plant pathology involves microbes like bacteria and fungi and viruses. But there were nematodes, which are animals. And if you recall, my love of biology was animal-based. So I thought, well, that's pretty cool. Let's give that a try. And probably one of the most important points of my life was he offered me 2 projects to work on cotton, one on soybean. And they all seemed fine to me. And I asked him which one would be better to work on. And he said, if you want to go into research, work on the soybean project. And that was the best single piece of advice anybody gave me. So I ended up getting a PhD working with soybean cyst nematode. As luck would have it, as I was graduating, Iowa State was looking to replace their longtime nematologist who had worked on corn nematodes. But soybean cyst had just shown up in the state in the late 1980s, late 1980s. They wanted a soybean cyst person, so it was perfect timing. I started on February 12th, 1990. And as they say, the rest is history.
For the uninitiated, we hear about, in the ag community, we hear about soybean cysts, particularly in the soybean part. But, nematodes are prevalent all over. There's, lots and lots of different kinds, right?
Yeah, so that's a good starting point. So nematodes are microscopic worms that live in the soil and in water. So ocean water has nematodes, pond water, river water has nematodes, lake water. And the soil is full of nematodes. Now, most of the nematodes that live in the soil are good nematodes. They break down organic matter, they cycle nutrients, and we want them in the soil for all the plant health benefits or soil health benefits. But as a plant pathologist that specializes in nematodes, I focus on the plant parasitic nematodes. And there are a bunch of those. There are a dozen or more species that feed on corn. Typically, they're not damaging. They're fairly common in Iowa fields, but every once in a while they can reach damaging levels, but it's not very common. But then soybean cyst nematode is, because of its biology, is one of the most damaging nematodes. And that's the one I get paid to work on to help Iowa soybean farmers produce soybeans profitably despite having soybean cyst nematode in their fields.
And you mentioned because of their biology, as I recall, that's one part of it. It's a particularly tricky version of nematodes. Why is that?
Yeah, so they are particularly tricky to deal with because even though they are microscopic worms that live in the soil, when the females develop, they lose their worm shape and they take on a swollen shape that really looks like a lemon, but it's microscopic. That adult female is about the size of a period at the end of a printed sentence in a book or a newspaper, and eventually it fills up with 250 eggs. So it has really, really high reproductive capabilities. And that happens in a matter of 30 days. The first females of the season might take 45 to 50 days because the soil is cooler. But then when we get into the rest of the season, it's cranking through a new generation every 30 days. So we can have 4, 5, or 6 generations in a single year. And if you start with the number 100 and multiply it by 250, which represents the number of eggs in a female, and do that 4 or 5 times, the numbers climb astronomically. So very high reproductive capability. But the second of 3 problems is how well it survives. So that lemon-shaped female that eventually dies, it's on the root, but the body wall becomes very tough and leathery, and that's when we start calling it a cyst instead of an adult female. It then is present in the soil full of eggs, and some of the eggs within inside that cyst or dead female can live 10 or more years without hatching. And without hatching, they're not vulnerable to starving or being eaten or so forth. So the nematode is extremely long-lived in the soil. And then the final aspect of its biology, and we don't understand why, that makes it really difficult to work with in terms of helping farmers manage SCN, is we can have 30% or more yield loss and the soybeans don't look sick. They don't look stunted, they don't look yellow, but at harvest time, there's 30% less beans. And so it's kind of a hidden pest, a silent killer. And when I speak with farmers, I liken it to high blood pressure. I say there's people walking around with high blood pressure that don't know they have it, but they could fall over dead at some point, although they probably don't. They just suffer chronic problems. And when it gets to managing it, they're told to do a bunch of different things. Don't reduce salt content, reduce alcohol consumption, take a medicine, exercise. And that is an analogy with soybean cyst nematode as well. Once farmers find out they have it, they have to take a broad approach to managing it. And it's a chronic health problem for soybeans that can be managed, but is probably never going to go away.
So you talked about, I don't know whether you say it's good timing or bad timing, but you kind of came in right at the point when soybean cyst was becoming known, right, as a pest. And I can remember back in the late 80s, I grew up not very far from the Stine farm there in central Iowa. And I can remember my grandfather coming home one day and saying, that Harry Stine had stopped by and had mentioned that that field must have soybean cyst, because I could see. And he'd never heard of it. It was like, well, I don't even know what that is. And so it's fascinating that was the cusp of that timing when we all hear about it now, but at the time it was very new and unusual.
Yeah, and your description of the timing is spot on. I started in 1990, but apparently there was a severe drought in the mid to late 80s. And soybean cyst nematode loves hot, dry conditions. And so that's when it first became apparent to Iowa farmers and to the Soybean Association, and then eventually communicated to Iowa State. And that coincided with my predecessor retiring. And I mentioned him earlier. His name was Dr. Don Norton, and he spent his entire career studying beneficial nematodes that live in the soil, as well as nematodes that feed on corn. When he retired, it lined up perfectly. Soybean cyst nematode was identified as a major threat to Iowa soybean farmers. And so the word soybean cyst nematode exists in my job description, which is incredibly unusual. All of my colleagues that work for other universities, their job description says they'll work on the biology and management of plant parasitic nematodes of important crops. My job description says that, but then there's a little phrase that says, with special emphasis on the soybean cyst nematode. So the growers recognize the threat. They talk to the dean of the College of Agriculture. And when my predecessor's position came up to refill, it was happenstance that I was ending my research in Georgia on soybean cyst and came here with the necessary tools to help farmers combat it.
So again, probably among certainly the leadership with the soybean checkoff, there was an understanding that hey, this is a problem and needs to be dealt with. But I can imagine, and I guess correct me if I'm wrong, that during those earliest days when you arrived, there was a huge gap in knowledge about what this was. I mean, I'm curious, what was it like in those earliest? Because part of your job is extension. I'm sure you were out talking to growers about this. What was that like?
Well, you're exactly right. It was interesting, and I was pretty much focusing while doing research to help come up with management options for farmers. My extension hat, I was just helping farmers figure out they had soybean cyst nematode and convince them that they had a problem, even though their soybeans didn't look sick. And that was a little bit challenging at first, being a new person around, new kid on the block in the 90s. But we worked at it. I've always had tremendous support from the soybean checkoff through the Iowa Soybean Association and another organization called the North Central Soybean Research Program. And over time, we kind of kept at it, and we convinced farmers to look for it. And a real turning point was a project funded by the North Central Soybean Research Program. So it wasn't just Iowa, it was 11 other states. And we created a project called the SCN Coalition. And we partnered with the universities in the region, the soybean checkoff organizations in the North Central region, and some private companies to get the message out to farmers about the threat of soybean cyst nematode and how to test for it. And it was one of the most fun things I've ever done. We hired a professional communications firm called Morgan Myers, based in Wisconsin. And they created a logo, and we did market research so we knew what farmers knew and what they didn't know about SCN. But the coolest thing for me was they came up with a catchphrase that summarized what we needed to do in 6 words. And that catchphrase was, take the test, beat the pest. Because in the 90s, farmers couldn't manage what they didn't have, or didn't know they have, rather. And so all we needed to do was help them figure out where they had soybean cyst nematode, and then they could start growing wonderful resistant soybean varieties and rotating with corn to manage it. But without knowing they had the pest, they couldn't manage it. So take the test, beat the pest was literally the perfect solution to our problem in the 90s.
Well, and that's a good point. As I recall, I think the checkoff board at that time was probably very keen on talking to you about what other solutions, what other things can we do, what do we need to do to battle this thing? And as I recall, I think to your point, the comment was, I just need to get people to do what we already know they need to do.
Right.
So it's more about communication.
Yep.
And we all know that, nobody likes to go looking for trouble, right? So if you can't see it, then don't tell me about it, because I don't want to know.
Yeah. Well, and having some geographical perspective, I came from Georgia. The year I left Georgia, it was after the 89 crop season, and their state average soybean yield was 27 bushels per acre. And they were doing press releases, how great that was. And then I move up here, and I hear people complaining back then in the 90s about 40 bushels, or not complaining about 40 bushels, 45. And that's always been a problem for us, is I think our soils are so productive and our environments are so conducive that we, without symptoms, don't know we're losing bushels per acre. And now I have documented data, farmers bringing in 55 bushels, and with using an unusual type of resistance called Peking, we can get 72 bushels. So when you have 55 or 60 bushels per acre, you're probably not bragging, but you're not complaining either. But we're leaving a lot of money out in the field in the form of bushels per acre we're not harvesting.
And that part of the story is probably where it intersects somewhat with, soybean breeding, right? I think in our industry, we know there's probably a vast majority of the material that's out there. Well, first off, there's two main sources of gene resistance. Right. Is that right?
That's exactly right.
So PI 88788.
Excellent. Yep.
And then Peking.
Exact.
In most cases, throughout most of our industry, PI 88788 is prevalent.
Right.
And from a breeder standpoint, they would tell you because that had more genetic potential than Peking.
It yields. If I can kind of play off of somebody's catchphrase.
Right.
It yields.
Exactly.
Yep.
Exactly. Now, what you said earlier, it also can hide a lot of ailments, right? If it already yields. But what's been fascinating to me is watch over the last few years in our lineup, and I don't follow other companies' lineups, but in our lineup, I'm starting to see increasingly more products come in our catalog that have Peking. I don't know. Is that similar to what you see other places?
Yes.
Okay.
Yes.
Because we used to have none, and now at least we have a handful,
Right.
and that's a sign that genetic potential is getting better. So we talked about this pest, and I don't know if the tactics have changed, but I guess as you talk to farmers, what are the things they need to be thinking about as they're thinking about SCN?
So this is a good time to introduce the second version of the SCN Coalition. So I mentioned it earlier in the 90s, and then basically we were on autopilot. Once farmers started testing for SCN, they grew these wonderful varieties with 88788 resistance. That got us through the 2000s and the 2010s, but there was a problem brewing literally underground, and it was the nematode was building up the ability to reproduce on 88788 resistance. So we launched in 2018 a new SCN Coalition with all North Central states, universities, and checkoff organizations, and some companies. And it's targeted at the more complicated situation that you just mentioned, and that is we have 88788 resistance. It's very common, but it's losing its effectiveness. We need to educate farmers about switching when they can to Peking resistance, being very selective in the 88788 resistant varieties they choose, because there are some really good ones and there's some really not good ones, and then rotating with corn. And we also have now a new tool in the toolbox in the form of seed treatments, nematode protectant seed treatments.
In addition to soil testing, right? What are things that they can do in season? Are there visual? What can they look for in their field?
There's some indirect symptoms that we hear farmers talk about, and we've documented them. And, I've been around enough decades that I've turned into an old storyteller. And one of my favorite stories is in the 90s, we at Iowa State had a research project funded by the Iowa Soybean Association, and it was at the very beginning of GPS technology. And so we were going to we did work with a leading farmer from Perry, Iowa. His name is Ron Heck, and he's a former ISA board member, former president of ISA, ASA board member, ASA president, just highly regarded. And he might have even challenged Iowa State to work on his farm. And he gave us 2 large fields to work in, and we gridded out 2 50-acre areas, one in each field, into 100 half-acre cells. And I still have a slide, a photographic slide, which I've scanned and digitized. The GPS unit, we had a staff member wear it. It was a backpack with a 4-foot antenna sticking out the back. So that was the early days of GPS. But a bunch of ISU researchers worked in Ron's fields, and we measured everything we could possibly measure, and that included soil insects, weeds, soil fertility, and of course, I was involved, so we're going to take soil cores and check for nematodes. And we discovered he had soybean cyst nematode, and he didn't know about it, but he was very quick to respond to it. One of the things we learned was we also had soil pH data, and the egg counts of the nematode numbers correlated almost in a straight line with pH. From lower numbers there were some half-acre cells that were 5.5 pH up to 8.0. And so that was a discovery that we went on to publish, and it was because of our work with Ron. But the thing that Ron observed, he was such a keen farmer and businessman, that the areas, when we showed him the maps of where we found SCN and what the numbers were, he said, well, that explains it. And I said, what? And he said, I have more weed problems in those areas. I have difficulty controlling weeds. And it kind of matches up with the areas of high egg counts. And what he was seeing was those plants weren't as thrifty. They weren't as big and bushy, and they weren't closing over. And anyone who tries to combat weeds knows that weeds love sunlight, and we get some control of our weeds once the rows close over. And so Ron immediately picked up on that. The other thing Ron did is, and I tell him every time I see him, he educated more farmers about soybean cyst nematode than I ever will, because he was a farmer leader, and he wasn't shy about talking about having SCN. And he told other farmers about what he found and how he managed it. And he's the very first person I know who grew Peking varieties in the early days and compared them with the 88788 varieties. But in addition to yield, he collected egg count data below ground by soil sampling. And he showed how it made dollars and cents in soybean production, not over 20 years, but over a simple turn or two of the corn-soybean rotation, by collecting his own data at that point and not being afraid to grow soybean varieties with this weird kind of resistance called Peking, which people have said, it doesn't have as high a yield potential as the common 88788 resistance, which is true. But Ron knew he was in it for the long haul, and eventually he got such good nematode control with Peking that it started to pay dividends in terms of yield. And number one, he was careful enough and meticulous enough to collect that data. And then number two, he wasn't bashful about sharing it with other farmers and with us to kind of magnify the message. So it was a really fun thing for me to do. And I stand by that statement that Ron has probably educated more farmers about SCN than me in our careers.
Great story. You mentioned that soybean cyst was confirmed in Japan.
Yep.
So it makes me curious. I mean, we always think about locally, but I mean, where in the world does this affliction occur?
Well, there's reports of some yellowish type disease in soybean fields in China in the 1800s. And so we actually think it originated in Asia, China or Japan. It was first found in a county along the Atlantic coast in North Carolina, and it was very near the coast, but it was a soybean production field, and it was near where ships would be brought in with ornamental plants from Asia, from Japan and China. So one hypothesis is that those plants that are brought in aren't completely clean of soil. In fact, if they didn't have any soil on them, they probably would have died during shipment. So they came with soil. And so that one hypothesis is that it was in the soil that was brought over with ornamental plants that were brought in the 50s. And again, it was found for the first time in 1954. The other hypothesis is that farmers and agronomists will know that soybean roots form these little nitrogen nodules, and it's bacteria that live in the soil that cause those little bumps on the roots, and those bumps are necessary for healthy soybeans because it takes nitrogen that's in the air and it permeates the soil and it traps that nitrogen and converts it into a form that the plants can use, soybean plants. That's called nitrogen fixation. And it takes these bacteria in the soil to attach to the soybean root and make that little nodule, is what they're called. Well, soybeans are an introduced crop to the United States. Some might not realize that. And when we first started growing soybeans, that bacteria was not present in the soil, so the roots weren't nodulating, and so they were suffering from nitrogen deficiency. Well, some brilliant, air quotes, brilliant scientists said, why don't we bring soil over from where soybeans have been growing in Japan, I forget if it's Japan or China, and sprinkle it in our fields to introduce the bacteria. And they did, but another hypothesis is that the nematode might have been introduced through that process as well. So that's how it got to the U.S. Within 4 or 5 years, it was found not only in North Carolina, but in the Boot Hill of Missouri. And I've not heard any stories of how that might have happened. But from the late 50s through the 60s, it spread then from the Boot Hill of Missouri up and down the Mississippi River. And one could say, well, maybe migratory waterfowl were moving soil, which had the nematode. And then it was found in extreme north central Iowa and south central Minnesota. Winnebago County, Iowa, and Faribault County, Minnesota touch each other in extreme north central Iowa. And in the early 90s, I heard farmers tell the story that somebody from that part of the world bought a piece of used farm equipment from the Boot Hill of Missouri and moved it up there. And within several years, they found soybean cyst nematode. The funny thing is, when they tell that story in Minnesota, they say the farmer lived in Iowa, and vice versa. When it's told in Iowa, the farmer lived in Minnesota. So we have no idea, even if it's true. But it underscores how this thing moves around. It can move around on anything that moves soil. And in the early days, that included farm equipment that was brought hundreds and hundreds of miles.
Yeah, because I recall in those earlier days of certainly of the coalition, they were talking about even things like if you found it in your fields, you should do things in a certain order, or don't take that equipment to a field that you don't know, that doesn't have it. Given the fact that soybean cyst has other plants that can serve as host, is it fair to assume that this pest is now prevalent pretty much everywhere soybeans are grown?
In general terms, yes, that's accurate. Being a scientist, I constantly want to refer to a paper or a set of data to answer almost any question. It's a curse. But in this case, we had funding from the North Central Soybean Research Program in the 90s to work with the U.S. Department of Agriculture's Ag Statistics Service. And we got a large grant to pay their people to sample randomly selected fields. Now, they're already sending staff to randomly selected fields, and they do this to this day to take plant measurements. It's called the Objective Yield Program. So when a farmer hears about yield estimates when we get to late season, it's not numbers just dreamt up or opinions. It's based on measurements that these personnel take in these randomly selected fields. We paid in 1995 and 96, we paid those people to pull soil samples from those randomly selected fields. And we actually did it for 6 states. But for Iowa, all the soil samples from all 6 states came to Iowa State. We processed the samples. We checked for soybean cyst nematode. And in Iowa, we found it in 70% of the fields. Okay, so that was in the mid-90s. And the real power about that is that it was randomly selected fields. And unless you're a statistics geek, it might not mean a lot to you that the power of randomly selected fields, but think about the power of randomly selected people. And up until, let's say, the last 5 or 10 years, even for national political campaigns, random surveys tended to be fairly accurate in predicting the outcome. So long term, scientists put a lot of emphasis on random selection. So that was the mid 90s. In the mid 2000s, I had enough money to do it again, but just for Iowa. And again, partnered with Ag Statistics Service in Des Moines. And we did it in, I can't remember if it was 06 and 07 or 05 and 06, 2 years using exact same methodology. But since they're randomly selected fields, they're different fields every year.
Sure.
The results were 72% of the fields. And that was in the from 2000 to 2010. The next decade, from 2010 to 2020, I got an itch to do it one more time. So in that decade, I did it again one more time with the U.S. Ag Statistics Service based in Des Moines, did 2 years of randomly selected fields, sampled and tested for SCN, 71% of the fields.
Really?
Yeah. So over 3 random sets of surveys spanning 20 years, every time we came in at 70% of the field. So it's not everywhere, but it's in a lot of places. And to go back to some of our initial comments, it's in places where farmers don't know they have it because the beans don't look sick. But it's out there in a lot more fields than people realize.
Yeah, it's interesting.
Yeah. Sorry for the long story there, but I've become a storyteller.
Yeah, that's fantastic.
Yeah.
So talk a minute about the seed treatment thing, because we talked earlier about, one of the unique facets of SCN is probably the cyst part of the component. And I know that's always been the tough thing to crack. So how does the seed treatment work to affect SCN?
Well, that's a complicated, or it's a simple question with a complicated answer, mainly because there's probably 10 seed treatments to pick from. Farmers might be most familiar with a couple different seed treatments that are chemical-based, that are offered by major seed companies and probably mid-sized seed companies as well, but there's biological seed treatments as well. And every single one of them has a different active ingredient and a different mode of action. So it is difficult to not become very complicated, or at least semi-complicated, in discussions of it. You'll notice, first off, that I didn't say nematicide seed treatments. I said nematode protectant seed treatments. And that's because scientists and university people get hung up on vocabulary all the time. But a nematicide is something that kills a nematode. The suffix side, C-I-D-E, means kill. Herbicide kills weeds. Insecticides kill insects. Nematicides kill nematodes. But many of these seed treatments do not kill the nematodes. What they do is they protect the roots from the nematode, either by repelling them from infecting the roots or by ramping up natural plant diseases to help the soybean roots better suppress the nematode reproduction once it gets into the roots. And again, with 8 or 10 different products with 8 or 10 different modes of action, it's hard to draw generalizations. But these things are out there. They, in my opinion, will not cut the mustard, if you will, on their own, but they're meant to be used with resistant soybean varieties as a way to shore up the performance of the resistant varieties.
But like you said, it's not about killing them. It's just about making the situation less favorable for them to cause the damage that they're bound to cause.
Right. And it may not offer protection throughout the growing season. That's a tall task to coat a seed, a tiny seed, and have it coated with whatever that's not nuclear to protect that root system for the entire growing season. So they are known to provide 4 to 8 weeks of protection. And that might sound troubling to a farmer or an agronomist, but it really isn't a problem. Back in the 70s and 80s, we had wonderful chemicals that we could they were mostly nematicides that we could apply to the soil. They were soil-applied nematicides. They're no longer available because of some environmental and human health reasons, but they worked. They worked well. But even then, they would protect the plants and control nematodes for a period of 4 or 8 or 10 weeks. Eventually, that wears off, that protection, and the nematodes that have survived it suddenly have a wonderfully healthy root system to reproduce for the latter part of the growing season. So I was taught in graduate school, soil-applied nematicides work, and there's published papers to show it. But at the end of the season, there often is as many nematodes as if you didn't use the nematicide, or there may be more nematodes because when you didn't use it, the roots were so unhealthy that it kind of held back the buildup of the nematode numbers over the season. So sorry for the long explanation, but a protection of the root system and the front end of the season for 4 or 6 or 8 weeks will pay dividends or can pay dividends in terms of a yield increase. But it's unreasonable to think they're going to provide long-term, season-long protection.
So just like everything else we do, whether it's weed control or anything, layered approach is probably the soundest practice.
Yes, we in the pest management world have a favorite phrase. It's called integrated pest management, and that's basically using everything you have available in a coordinated effort to provide maximum health of the plant and maximum yield and maximum suppression of the pest, in this case, the nematode.
So your lab, I know you do SCN testing for farmers, right? That they send in samples. I mean, a farmer who is interested can just submit a sample, you guys test it, send the results back, right?
Yes, it's the samples will go to the plant and insect diagnostic clinic. So if someone who's listening to this wants to hop online and check, if you just Googled ISU, maybe just the initials, PIDC, it might come up. If not, you'd have to type in plant and insect diagnostic clinic. And then nematode testing is an option there. And we, in my research lab, oversee kind of the processing and the advice part of the process. But we have a separate, nice facility that helps farmers as well as home gardeners and all Iowans have insect and plant disease problems diagnosed. And they are the ones who receive the samples, they process them, but the person who processes them earned his master's degree in our research program. We're right next door to each other. So we kind of rub shoulders. It's the plant and insect diagnostic clinic. Now, before we move on, I will mention there are some private soil laboratories in the state that also process samples for SCN. And the reason they do that now is because in the 90s, for the first SCN Coalition, one thing we realized was it wasn't the easiest thing in the world to have farmers submit soil samples for SCN testing. So we trained all the private labs we could get a hold of and that were willing to be trained in Iowa. So a farmer now doesn't necessarily have to take it or send it to Iowa State. They could send it to whatever laboratory they get their soil fertility samples processed as well. So there are some other options. And the easiest way I could tell people to see what are all the options for Iowa or any other state for that matter is to go to the new current SCN Coalition website. The website is thescncoalition.com with no spaces. Doesn't matter if you use capitals or not, thescncoalition.com. At the top of that webpage, there is a tab you click on for testing labs, and you click on it and it brings up a map of the United States. And when you click on Iowa, it brings up a pop-up box that lists our plant and insect diagnostic clinic, plus all the private laboratories in Iowa and surrounding states that will take Iowa samples. So we've tried to make it as easy as possible for Iowa farmers to find labs for testing.
Great. And you mentioned like with the SCN Coalition, that's really a broad coalition of your cohorts in other states, because of course we have customers and listeners in most of the Midwestern states. So I'm assuming they can go there and they can look up for their own state whatever the labs are that are cleared there.
Yep. And in addition, there's another map that they can find that shows the university expert in their state and that university's management recommendations, because we do vary a little bit from state to state, because North Dakota grows different varieties and has different cropping sequences than Iowa does. So a North Dakota farmer will need different advice in terms of long-term management than an Iowa farmer. So yeah, all that, we've made it applicable for farmers in every state that grows soybeans across the United States.
So we talked earlier about, some of the management practices being, planting resistant varieties, using seed treatments. I think we touched on rotation. Here in the state of Iowa, that's a little bit of a challenge because, we kind of have two favorite crops for the most part.
Yep.
Other states are a little easier, but that is a part of the conversation, right?
Yes, absolutely. I do not want to live in a world where we are not growing corn along with soybeans, because the only thing my colleagues in other states kind of give me the business, if you will, because the more non-host crops you can grow, the better. So, they say, well, I don't know how you can manage things when, you're growing soybean every other year. 1 year is like a nap for soybean cyst nematode, 1 year of corn. Well, that's not really true. We actually see great benefit of 1 year of corn. SCN egg numbers can drop up to 50% in 1 year of corn. And they drop some more in second year corn. But by the second year, you kind of lose your bang for your buck, is what I say. By third year corn, what's left in the soil are the dormant eggs that I mentioned earlier that can survive a decade or more. And so farmers that have really high egg counts, we may recommend that they grow a couple years of corn. And that used to be sound advice, but now with the high input costs for corn, actually farmers are doing the opposite and thinking about growing beans on beans. And that there's only thing worse than having soybeans grown every other year in the field, and that's having them grown every year from an SCN perspective. So
right.
That's not good.
I know at one point there was talk about, because you mentioned these cysts can overwinter and certainly last a number of years. There was talk about trying to figure out a way to sort of trick those cysts into hatching. Let's say you plant corn 1 year and you would spray the field with something. And is there anything ever come of that, or is it just challenging?
Yeah, well, I should thank you for making me talk about one of my professional failures. But, that's how research goes. It wouldn't be research if you knew something was going to work all the time. So you have a very good memory or very good knowledge. There is a natural chemical in kidney bean roots, and kidney beans are a host for soybean cyst nematode. And the Japanese scientists, they have SCN there, isolated the chemical that stimulated eggs to hatch. It's called glycinoclepin A. It's a really complicated molecule. So in the decade of the 90s, with soybean checkoff funding, I partnered with a chemist at Iowa State University. His name is Dr. George Krause, and we tried to synthesize that molecule or something that looked similar to it with the idea of coming up with a spray to put on a field when corn would be grown to trick those eggs into hatching. We had really good funding for, I think, three-year periods, and we just couldn't get it done. And we kind of probably were our own worst enemies, because we realized we were doing this to help farmers. So the chemist was only using reactions that could be upscaled or industrialized.
Sure.
And he was only using raw starting materials that would be affordable, because we realized we would need gallons of this to spray per acre, not little crystals or micrograms. And truth be told, before we started on our project, there was a chemist from Cornell University who made about a salt crystal's worth of this glycinoclepin A. But he used really refined, pure starting materials and reactions that you could never industrialize. So we started with some ground rules that really handicapped us, but we were in it to help the Iowa soybean farmer, and it wouldn't do us any good to make a couple crystals. We needed to make gallons, and we were never able to succeed. So after about 9 years, we realized it wasn't worth the funding of the soybean farmers, and we kind of discontinued that.
So what has changed over the last couple of decades about the approach?
I would guess the biggest change is we've fallen asleep at the wheel, being the farming community. We successfully managed SCN in the 90s and 2000s and 2010s and really didn't have to pay much attention, except for the nematode now has the upper hand on the PI 88788 resistant varieties. And so while I mentioned the SCN Coalition in the 90s created the catchphrase, take the test, beat the pest, we still use that because farmers to this day still remember it from the 90s. But our new emphasis is active management. And what that means is farmers need to become, or agronomists who advise farmers need to become actively engaged in managing SCN now. And that starts with sampling. Fall soil sampling to get a read on what the egg counts are, and then researching what are the best resistant soybeans in terms of yield and nematode control with the common resistance, PI 88788. What are varieties that are available for a farmer with the Peking resistance that they can grow in rotation with 88788 varieties? Certainly in that rotation, we want corn as well, and then the seed treatment. So it's just more effort to manage SCN now than 20 years ago, where it was as simple as take the test, beat the pest. Take the test, you're told you have cyst nematode, buy whatever resistant variety you want, your favorite seed company, whatever your agronomist advises is best for your field, and you were good to go. And that was literally true for a couple decades, but it's slipping away from us now.
So we talked about what's changed in the last 30 years and looking ahead for whether it's North Central Soybean Research Program or the SCN Coalition. What are the tactics and what are you hoping to do with growers in the next couple of years?
We hope to have them become more engaged in managing the nematode and to take advantage of all possible management options, which includes the two types of resistance. It includes non-host crops. And remember, the SCN Coalition works throughout the whole north central region. So there are states that grow more than just corn and soybeans. So that would be a big part of it. And then those seed treatments are a big thing as well. They all aren't going to give you identical results in any given field. And I and my colleagues at other universities can only do so much field experiments. We can't be in every field. And so I'm really starting to ratchet up my rhetoric, if you will. I don't know if there's such a thing as good rhetoric or bad rhetoric, but to have farmers become scientists. And I know they're scientists in other regards. They're very good scientists in comparing fertility regimes and tillage and effects of cover crops and other things. I think they need to do that with seed treatments and try a bunch of the seed treatments, or at least several, on their own and make careful observations and figure out which ones seem to work the best for them. And just in terms of yield, it hurts me to say this, but on record at least, that the nematode control is secondary for the farmer. So I want them to see what works best in terms of increasing their yields, but it has to be in bona fide nematode fields. Otherwise, you're studying something that's not there. It's like buying UFO insurance. And that's what brings us back to active management and sampling. So they know what's in those fields, and then they can start being more systematic in managing it, because the old tactic of take the test, beat the pest isn't cutting the mustard these days. It's slipping away from us by just knowing you have cyst and growing some resistant variety that you got from someone or someone's lineup.
So in addition to taking the test, it's about, like you said, active management, using all the resources that are available to you, right, and figuring out your plan of attack.
Yeah. And if a farmer's listening, it could sound overwhelming. Other than in science, I'm a pretty laid back person. And so it would be overwhelming to me if I wasn't a nematologist. But I would refer them to the agronomists. We have world-class agronomists to turn to, and they're constantly doing training, and they're up to date on our materials. Actually, the SCN Coalition website that I mentioned earlier has training materials for agronomists to use with their farmers, and agronomists are really quite capable and quite good at kind of taking all those management options and helping the farmers integrate it into a plan for long-term management. So farmers aren't alone, and they should take advantage of the professionals they have in the form of agronomists.
So you just mentioned the SCNcoalition.com, right?
Yep.
Other resources that if a grower wants to learn more, is there anything else out there for them to go?
Well, I have a website. I'm not sure how up-to-date it is, but it's called soybeancyst.info. So soybencyst.info. We try to put all our variety trial reports and lists of varieties and other things on there. There's some videos on how to dig roots and look for the little white females on the roots, how to pull soil samples. And then the North Central Soybean Research Program has a website as well, and we refer to it as SHRIN. It's the Soybean Research and Information Network, and it's a wonderful website, integrated many different soybean diseases and pests and fertility problems as well. It's funded by the United Soybean Board, which is the national organization, and executed by the North Central Soybean Research Program. But if a farmer or an agronomist would Google soybean and then SRIN, they would find a wonderful resource, a bunch of information, not just on soybean cyst, but on any soybean pest or pathogen or production problem as well. So those are three main places that are easy for farmers to go to.
Well, Greg, I appreciate you coming to visit with us today. The work you've done over the last, couple of decades has been fantastic on behalf of soybean growers. You and your counterparts, throughout the United States, really keeping growers aware of soybean cyst nematode. And as seed companies, hopefully we're doing our part to try to help those growers as well. But I appreciate you stopping in to visit today.
Oh, I love it. I thank you for having me. And I've got to tell you, I'm the luckiest person I know. I'm the envy of my colleagues to be having, number one, being in the state of Iowa as a soybean producing state. Number two, having the wonderful support of the soybean checkoff through the Soybean Association and NCSRP, and then having tremendous support from seed companies such as yours as well. I wouldn't do a thing different if I had to do it all over again.
Awesome. Well, thanks for being here.
Yep. Thanks for having me.
Well, that's our time for today. I want to thank our guests and our listeners for joining us on another episode of the Stine Seedcast. We'll be back again soon with more expert interviews and insights about all things Stine. To never miss an episode, subscribe to the Stine Seedcast wherever podcasts are found.
Subscribe to the Stine Seedcast wherever podcasts are found. To learn more about Stine and its elite corn and soybean genetics, visit stineseed.com. Stine has yield.